In this lesson, we're going to be talking about what it is that DNA is doing. So now we learned all about the DNA structure and how it's all put together and everything, and now we're going to talk about why it is that cells even have DNA, what its job is in the cell, and it's going to be kind of a complicated process. So bear with me, we're going to spend at least a week on this, so just get your notes done and then we'll talk about it in class. Just want to remind you that DNA is a double helix shape, and it's composed of nucleotides that are made of a sugar and a phosphate, and there are 4 different of these bases, and it's in 2 strands, 2 different sides that are complementary. Okay, they base pair up, adenine with thymine and guanine with cytosine. It's held together by these hydrogen bonds. So let's talk about what it is that DNA is doing. Well, the sequence or the order of the nucleotides on a DNA molecule is actually containing the code, the instructions to make a protein. Okay, so DNA is actually just containing instructions on how to build proteins. And proteins, of course, form the structures of your muscles and your bones. They form their enzymes that are doing important chemical reactions. They're just so important in every living thing. Most of what makes you who you are has to do with protein, and DNA is having the instructions for how to do that. So kind of the way it works is you have a DNA molecule, which is in the nucleus of your cells, and separate sections of the DNA are what we call genes that determine traits. So along a stretch of DNA, you might have a sequence of bases. A process called transcription copies the DNA onto another molecule called RNA, and RNA is also a nucleotide, but it's a nucleic acid, but it's a little bit different. And then from there, the RNA is translated into protein, and that's amino acids, a sequence of amino acids, as we talked about earlier in the year. So this is pretty much what DNA is doing. It's getting transcribed onto RNA and translated onto protein. So let's talk about transcription first. This is the process of copying the DNA. The RNA is the molecule that's going to carry the code out of the nucleus, because DNA cannot leave the nucleus. If DNA leaves the nucleus, it will be destroyed because there's too many chemical reactions happening in the cytoplasm. So DNA is protected. I say it's kind of like the king in its castle. The nucleus is the castle, the DNA is the king. It can't leave. So RNA is the copy that goes out of the nucleus to make the protein. RNA stands for ribonucleic acid instead of deoxyribonucleic acid. And RNA is different from DNA 3 ways. RNA is a single strand instead of a double strand. It has this sugar called ribose instead of deoxyribose. And it, instead of thymine, adenine pairs with uracil. It's another nucleotide base. It's just a slightly different structure. It's almost just like thymine, and it pairs with adenine. Now, there are 3 types of RNA that are involved in protein synthesis. The first we call messenger RNA, which carries the code, as I just talked about. There's also one called transfer RNA, or tRNA, and its job is to transport the amino acids. Okay, so it's transferring the amino acids. The third is called ribosomal RNA, or rRNA, which actually makes up the structure of the cell called the ribosome, which we talked about earlier in the year. And the ribosome is where the proteins are going to be made. That's kind of like the protein factory. Now, in the process of transcription, the first step is that the DNA has to come open. Okay? The DNA is 2 strands held together by hydrogen bonds, and so the cell has to go ahead and open up that DNA so the strands are separated, right there where the hydrogen bond is. Enzymes that are called RNA Polymerase are going to start the formation of the RNA. So you can see here it's opening up at this enzyme RNA polymerase. The DNA is opening up and it's copying a messenger RNA here. This new strand is forming along here. RNA nucleotides, adenine, cytosine, guanine, and uracil are going to be added according to base pairing rules. So adenine with uracil and guanine with cytosine. And that's happening here along where the RNA polymerase is, opens the DNA, and now it's making a copy of the messenger RNA. Notice where there's a T, there's an A, where there's an A, there's a U, a G with a C with a G, just like that. This process is called transcription. Now, the next part of the process is called translation, and we call it that because we're actually converting the message from one that's made of nucleotides to one that's made of amino acids. So it's a completely different kind of molecule. That's why we call it translation, just like when you translate a language. So once messenger RNA is made, now it's allowed to leave the nucleus. So if this were the nucleus of the cell where the mRNA is made, it's now coming out of the nucleus through this little pore, and it's going to where the ribosome is, okay? And it's going to attach to the ribosome to begin making a protein or polypeptide. This is kind of what that looks like. This is our ribosome. And remember, in the cell, these look like little polka dots that were on the ER. And where this ribosome is, this messenger RNA strand will attach so that it can begin translating. Now, in the cytoplasm, while the messenger RNA is being made, transfer RNA molecules, tRNA molecules, are already floating around, and they're attached to amino acids. Each one picks up a specific amino acid. Okay, so each tRNA is assigned to picking up one of the 20 amino acids. 20 different tRNAs, 20 different amino acids. Their job is to take the amino acid from where it's floating around to the ribosome where the protein is going to be made. And it's going to come in the order shown on the code on the messenger RNA. So this is a tRNA molecule. It is a nucleic acid like DNA. So it's got, in portions here, you can see double strands, and then it kind of curves around, but it's got some base pairing going on within. On one end, it has an amino acid attachment site. On the other end, it has something called an ANTICODON, which I'm going to talk about in a minute. Here's a simpler version of it. You have at the top a place for an amino acid to attach, and at the bottom something called the anticodon. Now, as the messenger RNA code is read, it's read 3 at a time, 3 nucleotides at a time, like CTA or GGA or CTC or AUG, something like that. Each 3 we call a codon. It's like a word. One codon is, think of it like a word. It's 3 nucleotides that make up kind of like a word. The transfer RNA has a sequence that matches called an anticodon that I talked about a minute ago. So if the codon is CCC, the anticodon would be GGG. It's the complementary base, the matching base. Okay? The anticodon then pairs up with codons that are on the messenger RNA. So let me show you what that looks like. So it looks something like this, where you have your ribosome, the messenger RNA with its code, and now we have these transfer RNAs coming in with attached amino acids. They have little partial words. This is phenylalanine, this is asparagine, I think, lysine, tryptophan. Okay, so these are different amino acids coming in attached to the tRNAs, and then on the other end we have UUU pairing up with AAA. We have CUA pairing up with GAU. That's kind of how it works. Now, there is a code. We call this the genetic code, and there's a code for every single of the 20 amino acids, and we have actually worked it all out so we know what the code is. And we use a table similar to this to tell. For instance, if we look at the first base in the codon, if it's U, we read this column. C is here. I'm sorry, that's a row. A is here, and G is here. And then the second base, we go down there here. So if it's UU, we're looking in this box. If it's UC, we're looking in this box. And then finally, for the third base, we look here. So UUU would be phenylalanine. UUC would be phenylalanine also. UUA would be leucine, and UUG would be leucine. Okay? So the way it works is there's a specific codon that signals the start of the process, and it's AUG. If you look at the chart a minute ago, AUG is for methionine, but it's also a start codon. So when the cell sees this AUG, it knows that's where it starts. At the AUG, tRNA molecules begin to bring in the amino acids. They read the sequence off the messenger RNA, 1 codon at a time, so 3 at a time, 3 bases at a time. And then there are special enzymes that glue the amino acids together into a chain that we call a protein. Okay, so let's look at this picture again. You can see here this chain up here, the enzymes are going to glue all these together. The next one's going to be this phenylalanine. It's going to get glued to the one next to it. Here's another diagram, just sometimes a different picture helps. Here we have our tRNAs coming in, bringing this little yellow dot, which is our amino acid. And here's the messenger RNA, and here's the ribosome. So these tRNAs are coming in. As they come in one at a time, there's an enzyme up here that's gluing them together, forming this long chain that we call a protein. Finally, at the end, there's something called a stop codon, which you saw in the genetic code a minute ago as well, and that signals the end of the gene. The finished protein is then detached. It comes off, and it floats away into the endoplasmic reticulum. So if you remember, endoplasmic reticulum has ribosomes. That's these pink things in this diagram. These pink things are the ribosomes. Here, this little string is the messenger RNA, and coming out the other end, this little string is the protein. So they're being made, and they're released into the endoplasmic reticulum. And if you remember earlier in the year, we talked about how the ER was responsible for processing the protein. And so they're going to go into here, into the ER, and they're going to be processed. And that's the end of basically the whole process. So from DNA, you have to make a copy onto RNA, which can leave the nucleus. It goes to the ribosome, where it's translated into protein. And that's the whole process.